Hello, my name is Kimberly Call, and I decided to go ahead and make a video for the antigen structure and antibody structure. This first diagram here is the antigen structure. The antigen binding site of an antibody or receptor on a lymphocyte has a unique structure that allows a complementary fit to some structural aspect of the specific antigen. This epitope binds to a specific TCR and is made functionally larger since it non-covalently associated with the MHC of the antigen-presenting cell. This epitope-MHC complex then binds with the TCR, forming a complex of TCR-MHC epitope. The interaction of an epitope with the TCR requires the APC to process that antigen. After enzymatic degradation takes place, the resulting small peptides associate with the MHC. Major classes of the antigens are carbohydrates, lipids, nucleic acids, and proteins. And of course here with this second diagram I did is the antibody structure. One of the major functions of the immune system is the production of soluble proteins that circulate freely and contribute specifically to immunity and protection against foreign materials. These proteins are called antibodies or immunoglobulins, which belong to the class of proteins called globulins because of their globular structure. Immunoglobulins can be membrane-bound or secreted. Membrane-bound antibody is present on the surface of B cells, where it serves as the antigen-specific receptor. Secreted antibodies are produced by plasma cells, then terminally differentiate B cells that serve as antibody factories and are housed mainly in the bone marrow. All antibodies share a common structure of 4 polypeptide chains, consisting of 2 identical light L chains and 2 identical heavy H chains. Each light chain is bound to its partner heavy chain by a disulfide bond between the corresponding cysteine residues, as well as by non-covalent interactions between the VH and VL domains and the CH1 and CL domains. These bonds enable the formation of a closely associated heteroimer. Multiple disulfide bridges link the 2 heavy chains together about halfway down their length, and the C-terminal parts of the 2 heavy chains also participate in non-covalent bonding interactions between corresponding domains. The antibody molecule form a Y shape with 2 identical antigen binding regions at the tips of the Y. Each antigen binding region is made of amino acids. The heavy and light chains both contribute 2 domains to each arm of the Y, with the non-antigen binding domain of each chain serving to extend the antigen binding arm. The base of the Y consists of the C-terminal domains of the antibody heavy chain. The overall structure of the antibody consists of 3 rather compact regions joined by a more flexible hinge region. The hinge region is very susceptible to proteolytic cleavage by the enzyme papain. Papain leaves the antibody at the hinge region, resulting in 2 identical fragments that each retain the antigen binding specificity of the original antibody. And the remaining region of non-binding portion, called the FC region, and also to conclude, 5 major antibody heavy chains are mu, gamma, delta, epsilon, and alpha. And I thank you for checking out my video.